Let us focus on the influence of pH in the molecular structure of amino acid. Under the normal physiological pH range, that is, the majority of human cells are said to be found within the level of 4, 5, 6, and 7. And in that situation, amino acid is said to be at its sweeter ion form. That means it has a dipole charge. The amine has a positive charge, and the carboxylic group is having a negative charge. However, changes sets in when the amino acid is subjected either to a very strong acidic environment or a very strong alkalinic environment. Now, let us observe as to what will happen to the amino acid if we will place it under a strong acidic environment within the level of pH 1. As you may note, the structural composition of amino acid under a strong acidic condition will change. The carboxylic group will be added with an additional hydrogen ion, and that the amine group will remain and retain its positive charge. And in this condition, we may state that the amino acid under a strong acid condition, under the pH of 1, is a positive amino acid, and that it is being protonated or added with a hydrogen at the carboxylic group. Now, as we move to the right of our pH scale, that is at a pH of 4, 5, 6, and 7, again, we mentioned that amino acid is at its sweeter ion forms, and that majority of the amino acid is within that pH level. Now, if we will subject the amino acid into an extreme alkalinic environment, that is at a pH of 9 onwards, let us see as to what will happen to the amino acid molecule. At the pH of 9, a structural changes in the amine group will occur, and that is the hydrogen will be reduced, and that is a condition which we call deprotonation. And the carboxylic group will retain and remain its negative charge. And so therefore, the amino acid subjected to an extreme alkaline environment such as 9 and above will become deprotonated, and the carboxylic group will retain its negative charge. Peptide bond formation. It refers to a chemical bond that is formed between two molecules of amino acid. This discussion will answer the question of what connects one molecule of amino acid to another, and another, and another, until it creates a polypeptide of many to hundreds to thousands connected amino acid. Below is an illustration sample of selected amino acid that will bind with their chemical structure, namely the proline, the phenylalanine, the lysine, and the tryptophan. With a keen look, at the left, we may notice that the left side of each amino acid structure is composed of an amine group, that is the H3N positive. And this is bound to connect to the right side of the carboxylic group through a particular process. And we call this process as dehydration synthesis or the condensation reaction. This happens when one amino acid carboxylic end reacts with the other amine ends of the amino acid. And this will form a special covalent bond known as the peptide bond. And even more, it will cause a reaction between the hydroxylic group or the hydroxyl and another hydrogen, which further will result to the release of water as an end product. Now, this combination of a peptide always results to a formation of a short to a long to a very long chain, which we designate the left side of a long chain as the N-terminus, and we can designate the right chain as the C-terminus. Finally, when we combine two short amino acid, then we call that as dipeptide. And when 3 to 10 amino acids being combined, then we can call it as oligopeptide. And when a chain of 100 to 1,000 of a complex molecule of protein is being combined, then we will call it as a polypeptide chain. The protein structure. It refers to the orientation and arrangement of the subunits of a protein, even the multi-subunits. Now, protein structure will try to fold into a specific shape, and then it will carry on with its sequence of amino acid from a monomer to a polymer, and this folding and shapes of a protein structure will determine the function as it is related to the shape and size of a protein. There are 4 basic structures of the protein. And number 1 is what we call the primary structure protein. By the looks of it, these are achieved by combining an amino acid of different types. Second to that is the secondary structure. Now, from the secondary structure, there will be folding of itself, and voila, a form is being created. And the third form, it is even more complicated and folded, and that is what we call the tertiary structure, as it folds itself. And the fourth type is an even more intricate and complicated type combination, and this is what we call the quaternary structure. Now, in the next slide, we will try to describe it even more. Let us focus on the primary structure. It refers to the orders of amino acid in a peptide chain, the linear sequence as encoded by the DNA, as you may look at it. They are joined by a peptide bond, and they are linked. And as you may notice here, the water is being released for each time the peptide bond is being formed. Now, the link series of the carbon, the nitrogen, and the oxygen atom is the secondary structure. It is folded into 2 types, the alpha helix and the beta sheet. The alpha is coiled as a spring, and this is being stabilized by a hydrogen bond between amine and carboxyl group. Now, beta sheet is folded when hydrogen bond is stabilized by 2 or more adjacent structure and held by hydrogen. The tertiary structure. This refers to the overall shape of a polypeptide protein chain. And once they twist upon themselves, they are now called as a protein. And most of the protein are in a globular shape, one that is being shown. That is with a hydrophobic side chain. The quaternary structure. This refers to the interaction between a multiple protein and a polypeptide chain. They come together, as you may notice there are 4 complex protein, to cooperate to form a complex protein to pick up oxygen. And the most common example is the hemoglobin in the RBC and the sodium channel in cell membrane. The protein representation, it refers to the size and shape of a protein as a clue to the function of each protein molecule. And at the right is a ribbon diagram or a cartoon diagram showing the organization of protein backbone and highlights the helices. Another is a surface representation that shows the areas accessible to water molecule. Now, protein size can be as small as 6.5 nanometers, similar to the wavelength of light. And the high concentration of hemoglobin can be 100 nanometers in size within a 1 RBC that can contain 280 million of hemoglobin molecules. Once again, you may pause the video. Let us assess and check your comprehension. That's all, folks. This has been your Nurse Radiologico Guru saying thank you for watching and enjoy studying.